In this lecture, we're going to be talking about the last macromolecule, lipids. So lipids are my favorite subject. There are many types of lipids and many things that we can talk about, but in this lecture or combined lecture, we will primarily focus on what lipids are, what they're made of, the different categories of lipids, and go straight into lipid membranes. So what the structure and function of membranes are, and how we can finally be able to apply some physics to check out the diffusion of different ions and molecules across the lipid membrane. After going through a recap of nucleic acids, we'll start with defining what the building blocks of lipids are, which are fatty acids, and defining what types of lipids there are can get very complicated depending on how you're looking at categorizing them. So essentially, I'm going to go through 8 categories of lipids that are generally accepted. So going through that, you'll be able to gain a deeper understanding of just how many lipids there can be. And then by that, we'll understand that lipid membranes aren't just made of one type of lipid, but can be made of many, many different types of lipids. So we'll be taking a look at what biomembranes are, their structure and function, and taking a look at different parts of the membrane because it is asymmetrical, because it's made of different components of lipids in the top and the bottom leaflet. So it gives rise to different domains or rafts within your lipid membrane that can carry out specific functions, especially because those lipid rafts and domains are usually very rich in different types of proteins. So usually we can say, oh, this lipid raft has a specific function because of the certain proteins that are there. Afterwards, we'll take a look at the biophysics of lipid membranes. So this is the only lecture for macromolecules where we're actually applying some of the equations that we learned before to a biological topic. So what we're going to do is talk about the transport of different ions or different small molecules through the membrane, and they can happen through passive or active transport. Specifically for passive transport, you can go through a gradient. So if you have a highly concentrated area, you're going to go towards a lower area of concentration. So that's going to be based on Fick's law, which we've talked about before. So we'll be able to create an equation for that. Apart from that, because of the fact that you can have more ions on one side of the membrane than the other, you generate a membrane potential. And because of a membrane potential, you also can create another gradient, an electrochemical gradient that we can look at through Fick's law as well. So let's start with that recap of nucleic acids. We know that nucleic acids are made from nucleotides, and those nucleotides are going to be connected together through a sugar-phosphate backbone or phosphodiester bonds. After that, you can also have glycosidic bonds that are attaching these bases to the sugar phosphate backbone, and you have 4 different bases, 2 pyrimidines and 2 purines. So you have AT, that are 2 bases that are going to have 2 hydrogen bonds between the 2 of them, and you have C and G, which have 3 hydrogen bonds between them. Because of the different number of hydrogen bonds, you therefore have AT base pairs and CG base pairs. And so this alternating or random sequence of these base pairs is going to create a DNA double helix. What's important to remember is that your DNA helix, the ones that we have, the double helix, is in a B form, and that means there's 10 base pairs per turn. There are 2 other forms of DNA, the A form and the Z form. The A form has 11 pairs per turn, and the Z form has 12 pairs per turn. So going from DNA to protein, that was the majority of our focus in the previous lecture. DNA is going to contain instructions for making a protein through an intricate number of steps. So we're not showing DNA replication here because that's just creating an extra set of DNA. But if we're really trying to create protein from an existing strand of DNA, it's going to be unwound, and a pre-mRNA transcript is going to be created from the unwound DNA. That pre-mRNA transcript is going to get processed. First, it's going to be protected on each end. There's going to be a 5' cap and a poly A tail, 3' poly A tail. Apart from that, you can kind of see over here that there's a shaded out version. We're just going to assume that's an intron and it's an interfering codon. So we splice out all of those interfering codons and we get, and we only keep the exons. So keeping those exons, we end up with our mature mRNA transcript, and that is like the transcript that your ribosomal subunit will use to form that initiation complex and create a growing protein chain. So that's the translation step. What's going to happen is your ribosomal subunit is going to come together, create that initiation complex, and you have transfer RNAs that are going to bring in amino acids that match with the tRNA is going to bring in amino acids for specific codons. So the tRNA will bind to the codon that its anticodon matches with and continuously help form these peptide bonds for a growing protein chain. Eventually, stop codon is going to be reached, and in the exit site, the entire protein will get released. The entire initiation complex will be dismantled, and you end up with your finalized protein. Now, we also talked about genetic mutations and disorders. They can come from single or multiple gene mutations. For single gene disorders, we took a look at sickle cell anemia. That means we have hemoglobin S instead of hemoglobin A. Forgot the A there. So hemoglobin S is going to take on a sickle shape. Basically like a curve, and it causes the stacking and sticking of red blood cells in your blood vessels, which causes a whole bunch of problems, primarily the blockage of blood flow in your veins or your blood vessels, which means that the essential oxygen and nutrients that are supposed to go to your tissues will never get there, and so it creates a lot of cell death. You can have chromosomal disorders like Down syndrome, which we talked about, basically an extra chromosome 21. You also can have complex disorders, which is multiple mutations in multiple genes. One of them is colon cancer. Usually you have mutations in FAP and HNPCC genes, which almost guarantees that you'll get it if you do have any of these inherited mutations. We can now move on to lipids. So we first want to talk about how lipids are even created. What are they made of? And the general consensus is that fatty acids are the main constituents of lipids, or the fundamental building blocks of lipids. The only problem is that lipids are quite complex. So in terms of fatty acids, there are many different types and many different attachments that we can consider. So we'll take a look at a fatty acid and common lipid structures, then take a look at the categories of lipids as well as their functions, then take a look at specifically phospholipids and the cell membrane. So what are lipids made of? Lipids are macromolecules that are soluble in nonpolar solvents. So both of them are nonpolar and they're going to like each other. So unlike other macromolecules, there really is no true monomer for lipids. It's not like amino acids and proteins and nucleotides for nucleic acids. Lipids are made up of many small and non-repeating components. So we usually refer to fatty acids as the main building block of lipids, but we have to define what fatty acids are first. So a fatty acid, the example is here, and there's two types. They are chains of hydrocarbons, so basically a whole bunch of carbons with hydrogen attached to them with various lengths. So you have to have at least four in a chain to be considered a fatty acid. You also have different levels of saturation, and saturation is an important term to know. If you have a highly saturated fatty acid, it means that you have a bunch of hydrogens in your carbon chain. So if it's fully saturated, it means that all of your carbons have hydrogen bonded to them and nothing else. If you have an unsaturated fatty acid, it means that you no longer have hydrogen bound everywhere. You have these kinks that are introduced. So a double bond is introduced between two carbons, which means you've lost hydrogen on one side, and it creates a kink in your hydrocarbon chain. So as you can imagine, if you have very linear tails, they're going to be longer, but if you have more kinks, your tail is going to be a little bit shorter if you have the same length of a hydrocarbon. So different types of fatty acids with different amounts of saturation are going to affect the length of the tail and also the different interactions that it can have. Fatty acids also require a glycerol to become lipids. So if we have our fatty acid, we still have to consider the attachment of glycerol so that it can become a lipid. So how do we form a lipid molecule? Lipids are going to be formed when the alcohol of your fatty acids, because you have a carboxyl group there, and a group of glycerol is going to esterify together. So it's going to go through a dehydration synthesis. And then you end up with a true lipid. A lipid cell is usually called a fat cell. And in terms of categorizing fatty acids, sometimes fatty acids are used on a standalone basis. We consider those to be a type of lipid as well. So some common structures that we are going to take a look at are phospholipids, sterols or steroids, and sphingolipids. So these are three of the most common ones. I'm going to be going through all 8 of them. So what are some common lipid structures? In most cases, you'll find lipids with a hydrocarbon tail, and these are nonpolar. They're nonpolar, they're hydrophobic or water-fearing, but usually their heads, whatever they are, they're hydrophilic or polar. And that means that they have a hydrophobic region and a hydrophilic region. And so we call a lot of these lipids, specifically phospholipids, amphiphilic, which means they have properties of water-loving and water-fearing components. Now, the 8 categories of lipids, you should be able to understand the differences between them, and I'll point out which ones are the most important to know. The first category of lipid is just a general term because all lipids are made of fatty acids, so we call standalone fatty acids a separate group. Fatty acids are groups of molecules with hydrocarbon chains that terminate with a carboxylic acid group. Because fatty acids can be their own standalone group, or they can be considered building blocks of lipids. If they are considered as building blocks of lipids, then we call them fatty acid residues. If we say fatty acid, it means that we're taking a look at a standalone fatty acid without it becoming a lipid. Any fatty acid that has a double bond can exist in a cis or trans form. So this is very important. If it's a cis form, it means that both of the both of the hydrogens are going to be in the same direction. If you have a trans double bond, the H's are going to be on opposite sides of the double bond. So in most cases, you do need a cis double bond to create that kink. Next, you have glycerolipids. Basically, they are composed of substituted glycerols. So the best example is triglyceride, which is the example I have here for the structure. Triglyceride, basically you have three groups of glycerol. So the three hydroxyl groups of glycerol are going to be esterified to your three fatty acids. The three fatty acids that are connected can definitely be different. So for triglyceride, you have palmitic, oleic, and alpha-linolenic acids. The ester bonds, in terms of these triglycerides, it's these ester bonds over here that get hydrolyzed when we metabolize fat cells from our fat tissue. So back then, we talked about the main functions of different macromolecules. Lipid wasn't just a protective barrier, it also provided long-term energy storage. So if we do need to use lipids, so basically for fat burning purposes, we get to hydrolyze the ester bonds and get the energy from that. The next category is sphingolipids. So sphingolipids are a family of lipids that share a sphingosine-based backbone. Sphingosine is the major sphingosine base of mammals. So you don't have to know too much about this, but there is a very common sphingolipid called sphingomyelin. Its official name is ceramide phosphocholine, and it is a major sphingolipid of mammals. It is actually found in the myelin sheath surrounding the axons in your neurons, and structure looks like this. I want to explain what the different parts of the structure were. So in black, you have the sphingosine, which is the main backbone of your sphingomyelin. In red, you have phosphocholine, and then in blue, you have your fatty acid. Next, you have steroids. Basically, one H is going to be substituted with an OH group at position 3 of your hydrocarbon chain. The most important steroid that we're going to be looking at is cholesterol, extremely important in the cell membrane. Cholesterol and its derivatives are going to help maintain membrane fluidity in the fluid mosaic model of the membrane, and they're usually found embedded in the membrane. So as you can see over here, if you have two phospholipids, your cholesterol is going to be embedded in between the two lipids, and it provides additional support and structure. So the rigidity and the elasticity of your membrane are going to be highly dependent on how many components you have inside your membranes. So cholesterol can stiffen a region, especially in the middle because it's more compact, and the areas where there's no integration are going to be a little bit more fluid. Apart from that, cholesterol also has some cell signaling roles. Next, we can take a look at prenols. This I'm not as familiar with, but they are synthesized from 5-carbon precursors via a MVA pathway. The prenols are really important in the processing of proteins, carotenoids, and fat-soluble vitamins. And it's I've just presented an example of a prenol, which is geraniol. And in that case, all I'll say here is that prenols do play a role in vitamins, and that would basically be it. For sacharolipids, the name itself tells us that it looks like it's a fatty acid with some sort of sugar or carbohydrate backbone. So one common saccharilipid is glucosamine, and glucosamine is really important when we take a look at bacterial cells. So 2 glucosamines are actually found in lipid A, which is a lipid component of the LPS lipopolysaccharide layer of a gram-negative bacteria. So gram-negative bacteria don't just have an inner membrane, they have an outer membrane as well. So in the outer membrane, there's this lipopolysaccharide layer. So it's got like many tails coming out. And this lipid A is some sort of toxin. So lipid A is going to have a core polysaccharide and an O polysaccharide, and together it forms the lipopolysaccharide layer of your outer membrane of a gram-negative bacteria. Next, you have polyketides. These are lipids that contain a lot of secondary metabolites and natural products from living sources. And so they have a lot of structural diversity. The most important thing you should know about polyketides are that they're primarily cyclic, and that most antimicrobial agents are this category or derivatives. Tetracycline, very popular antibiotic, is an example of a polyketide. And tetracycline, many types of antimicrobial agents are actually derived from different bacteria. So tetracycline is an antibiotic that comes from the Streptomyces bacteria, which I studied for my PhD. Not tetracycline, but another antibiotic coming from Streptomyces. Is comprised of 4 different rings, and from all of these backbones, you can make a whole bunch of modifications to their backbones through different chemical reactions and processes. It doesn't really change their antibacterial activity, but rather their half-life and binding affinity. So in many cases, with polyketides being antibiotic agents or antimicrobial agents, you can modify these backbones to help them be more attracted to a specific target. So that's where that comes in. The last one I want to talk about is the most important one, because I wanted to end off here so we can continue our discussions about phospholipids. Phospholipids are 2 hydrophobic tails and a hydrophilic head with a phosphate group joined by glycerol. So the key components of all biological membranes is a phospholipid because of its amphiphilic nature. So what are the main functions of lipids? I'm really just going to go through just general functions. So there's structural support, mainly from phospholipids forming biological membranes. And those biological membranes are going to be formed by the hydrophobic effect. You have energy storage. So like I talked about before with fatty acids, your fat cells, adipocytes, are going to continuously synthesize and break down triglycerides. You also have cell signaling mechanisms where you have lipid messengers binding to protein targets. In terms of biological membranes, we're going to dedicate the entire next section to this. Basically, because of the hydrophobic effect, let's say you have your lipid bilayer here. Number one, because of the hydrophobic effect, and because of the fact that your phospholipids are amphiphils, you have water surrounding all of your lipids. The polar-loving or the water-loving components of the phospholipids are the heads. So the heads are going to want to face water. So they're going to line up here to face the water on top, but there's also water on bottom. So that means a second layer of lipids have to come in so that the tails that are hydrophobic are protected from water, and you end up with 2 layers of heads facing the water. So schematically, it looks like you have a linear layer of polar heads and a linear layer of a hydrophobic core. But in real life, that doesn't really happen. There's going to be what's called edge effects. So this isn't energetically favorable. What's going to happen is these hydrophobic tails are still exposed at the edge of the sheet. So they're going to want to come together and form a vesicle, a completely closed compartment that shields all of the tails from water. So that is why a lot of our biological membranes are in the shapes of completely closed surfaces. So now we're on to lipid membranes. First, I want to talk about the structure and composition of lipid membranes. The fact that they really aren't just lipids; they're way more than that. We'll take a look at the fluid mosaic model, which explains how lipids can move around throughout each of their layers. Talk about specific regions of lipids that we can localize, and then take a look at different proteins that are embedded in membranes. So when we talk about lipid bilayers in cell membranes, we have to know that it's made up of two monolayers of lipids. So one layer at the top, one layer at the bottom. It's never going to be planar. It's going to be in the shape of a closed surface. You have hydrophilic heads pointing towards the aqueous environment and hydrophobic tails of lipids creating a hydrophobic core. The thickness of the core is usually 5 to 10 nanometers. We say 5 nanometers because it's the minimum, but it's going to be determined by how many kinks are in the tails and how long the hydrocarbon chains are. But in general, you have 5 to 10 nanometers. Not only that, you have a bunch of proteins that are embedded in your membrane. It doesn't have to be just proteins. It can also be proteins attached to different carbohydrates or other components. So in a more detailed look at the cell membrane, a generally and widely accepted model of the different movement of or the different properties of cell membranes is the fluid mosaic model. So what the fluid mosaic model explains is that, number one, your lipid membrane isn't just made up of lipids, it's made up of many different things. You can have simple proteins attached to your membrane, or you can have different surface and integral proteins partially integrated or just on the surface of your lipid membrane. You can also have different carbohydrates, or you can also have protein channels. Most importantly, in terms of the cytoplasm side of things, there is usually an extracellular matrix that's represented here that helps keep it stable. So that is usually some sort of cytoskeleton that prevents it from moving too much, or else it could literally just go anywhere. So the other thing that the fluid mosaic model explains is how lipids move within the membrane. So looking back at that picture from before, there are so many components to the lipid membrane. What if we want one component to move elsewhere? So the important thing is that if you have a bunch of proteins embedded in your lipids and all of the protein and all of your lipids can kind of shift around, then that means that your lipids can move in several different methods. Number one, your lipids can move laterally throughout your membrane, and that is called lateral diffusion. You can have your lipids, so looking at the individual lipid, it can twist and turn. So that means your phospholipids can rotate or flex their tails to accommodate for the insertion of proteins or the insertion of different molecules like cholesterol. There's a really rare event that happens, but is very, very common in drug metabolism, and that's lipid flip-flop. So in lipid flip-flop, what happens is one of your lipids just decides to flop over to the other side. And a good example of this is when you're creating a membrane pore. So usually in most types of antibiotics or drugs that you take to help cure something, you're trying to create a membrane pore in your bacterial membrane so that ions can leak out of the cell and kill the cell. So in some cases, you end up with your antibiotic grouping together to form some sort of cluster on top of your membrane. So let me just draw this. So this would be your lipid cells, or phospholipids. And there's that cluster of antibiotics. In some cases, because it's possible to have lipid flip-flop, this whole purple thing flip-flops to the other side along with the lipids. Not the greatest drawing, but now you have two chances for a pore or a cluster of antibiotics at the top and a cluster of antibiotics at the bottom to come together and merge and bind so that you create a membrane pore. In that case, you end up with an ion channel that will do its work and kill the bacteria. So there are many different mechanisms of action for different antibiotics and drugs, but that's just one of them that really takes advantage of lipid flip-flop. Next, I want to talk about membrane asymmetry. So cell membranes exhibit membrane asymmetry, and what this means is that lipids are always different. Lipids and proteins on either side of the membrane are not symmetrical. It's not like you have, let's say, phosphatidylcholine at the top exactly at this location, and there's another phosphatidylcholine at the bottom. Because of the lateral diffusion of proteins and lipids on each layer of the membrane, it's very asymmetrical. And asymmetry is required for two very important things. Number one, the ability for regulating the transport of molecules from one side of the membrane to the other, as well as the fact that you can create an electrostatic potential difference between the two membranes. So maybe one side has more positive ions and the other side has more negative ions. So the other thing about asymmetry is that it allows for lipid domains. When we talk about lipid domains, which I find are really, really cool, lipid domains are certain areas of your lipid, and it's just lipids. We don't take a look at anything else, like proteins or anything. We're taking a look at just the lipids. A lipid domain is an area of the lipid that is highly concentrated in one type of lipid. So let's say this is lipid number 1, this is lipid number 2, and lipid number 3. They've got different tail lengths and different similar characteristics. Each of these are individual domains, just in this example. It means that my red lipids are all grouped together, my blue lipids are all grouped together, and they form their own domain of lipids. That means they're going to exhibit certain properties, and maybe they're going to attract certain proteins to bind to them. So areas that are rich in specific lipids may indicate a localization of certain processes that take place. Because, for example, if you have a highly electronegative region in the blue area over here, maybe a protein binds to it. So then, understanding that, we move on to something called lipid rafts. Because of all these lipid domains, you can have lipid rafts within the membrane. So that means maybe you now have a transmembrane protein that's highly attracted to that blue area, just as an example. So then they attach always to that lipid domain, and you now have what's called a lipid raft. So if you have a collection of different proteins, different cholesterol, sphingomyelin, and different proteins that help stabilize those transmembrane proteins as well as the lipids in that area, you end up with a very rigid component to your membrane. And so those are called lipid rafts because they're very specific to a process, because it's specific to a certain protein or enzyme. And not only that, because they're very rigid due to cholesterol and they're stuck together, they move as a raft throughout the membrane. So a lipid raft is specific in its function and also its movement. So different types of proteins in the membrane. Because we know that there are lipid rafts, it means we know we can study the effects of different proteins in certain areas of the membrane. There are different types. There are transmembrane proteins, which is what we're going to focus on. There are monolayer associated proteins, which means that the protein is only attached to one side of the membrane. You can have lipid-linked proteins, which means it's a lipid, and then you have some sort of carbohydrate or something attached to it, and then another protein attached to that. So it's almost like it's being anchored to the membrane in some fashion. And of course, you can have protein-attached proteins. So maybe this protein, transmembrane protein, is attached to another peripheral membrane protein. So now I want to focus on transmembrane proteins specifically. Transmembrane proteins, there are essentially 6 main types. You do not have to know a lot of details about them, just know that there are different types and different functions for each. So a receptor protein is a transmembrane protein that usually is going to attract and bind a chemical messenger to pass on a signal. An enzyme is going to also have an active site or a binding site for a chemical messenger, and it'll fulfill its process, maybe break down a chemical, speed up a reaction, and then afterwards your chemical messenger is either disintegrated or disassembled or repurposed. You can have different ion channels. So these two are ion channels, allowing ions to go back and forth between one side of the membrane to another. Here is just a channel. It's constantly open. It allows for a free flow of ions going both ways. Over here is a gated channel. So it's a gate that allows ions to go through if it's open, and of course it blocks it when it's closed. So usually these are moderated by other enzymes and components that are attached to the protein so that it can be turned on and off. You have cell identity markers, which are proteins with carbohydrate attachments. Essentially, what we're looking at here is a glycoprotein. It has different surface components that will allow the body to recognize it or not recognize it as part of the host or an invading cell. These are cell adhesion molecules. So basically, if you want several cells to stick together, these can signal to other cells and help anchor them to the same spot. Now let's take a look at the biophysics of lipid membranes. So finally, we get to take a look at applying some of our physics knowledge to lipid membranes. So the first thing we want to talk about is membrane permeability. The cell membrane, very important to know that it's semi-permeable. It's only going to allow certain molecules or ions to pass through it. And so looking at this picture here, you have some ions, for example, that can't go through, so they're bounced back. So that means they have to stay on that side of the membrane. Large molecules, obviously, the larger it is, the less of a chance it has to pass through. You have small nonpolar molecules that easily diffuse through. And the reason for that is because they are nonpolar. They can go through, they are going to be okay passing through the hydrophobic core and then pop out to the other side. Small polar molecules will require a little bit of help unless you have an ion channel or a concentration gradient. So because of the fact that some ions can go through and some ions can't, or molecules, it means that usually there's one side of the membrane that is more positive than the other, or vice versa, and that means it gives rise to a membrane potential. So here, a membrane potential is created by the different concentrations of ions on opposite sides of your cell membrane, so one on each side. The typical values are -40 to -80 millivolts for the exterior of the cell. So over here, the exterior. The most common ions that have a concentration gradient are sodium, potassium, and chloride. And so over here in this picture, if the membrane is selectively permeable to potassium, for example, It means that the potassium, these positively charged ions, can diffuse through your ion channel, leaving behind a bunch of A's. So those A's are large solutes that cannot pass through the ion channel or the membrane. So that means if your potassium ions are going through to the other side, then the right area is getting more and more negatively charged. So this separation of charge creates a membrane potential. And now our question is, how can they move across the membrane? We now have to take a look at different types of transport. So membrane transport can be passive or active. Passive means it doesn't require energy, whereas active means it does. Passive transport relies on the second law of thermodynamics, basically the increase of entropy, and follows Fick's first law. So combining the two of these laws together, it means that your ions are going to move from an area of higher to lower concentration to increase entropy. So that's very, very important to remember from a physical standpoint. There are various different ways of passive transport. There's simple diffusion, facilitated diffusion, filtration, and osmosis. Now, with active transport, usually you have to use metabolic energy to transport molecules across the membrane. So usually you have to require ion pumps and channels to cross. Now, we don't have any examples for this lecture because when you start applying equations to biological systems, they can get fairly complicated. But what we can do is present a model of a mathematical model of how to represent passive transport. So passive transport is really just diffusion. It requires a gradient for the molecules to move across the membrane to reach equilibrium. So the only thing with membranes is that the gradient can be based on a concentration gradient or an electric potential gradient. So using Fick's law, we have to create two different forms of Fick's law. The first form of Fick's law just has to do with the concentration gradient. So that's the one that we're familiar with in red. In purple, we have a modified version of Fick's law for electric potential. So you now have the concentration on a standalone basis. You have the charge Z of one ion. Maybe you have a sodium ion, which is a +1 charge. You have F for Faraday's constant, and you have R and T for your temperature and for your gas constant as well. Now you have this, which is your electrochemical potential gradient. So that's just a different gradient, but you also apply Fick's law. Now, if I'm taking a look at passive transport as a whole, I have to take a look at the total amount of flux coming from the concentration gradient and the electric potential gradient. So that means I just add them together. In blue, I have my total flux is equal to the two of these added together, which is equal to this. So if you combine these two equations together, this equation in blue is called the Nernst equation for one diffusing ion. So that's also a very famous equation, and it involves the combination of two fluxes from two gradients. Now, there are also different types of active transport. There are two types we have to look at: primary active transport and secondary active transport. Primary active transport requires the hydrolysis of ATP, and that hydrolysis of ATP is going to phosphorylate your transport protein. Basically, it turns it on. It's going to cause a conformational change like this, like the sodium-potassium pump here, and then allow 3 intracellular Na ions to go to the other side and 2 potassium ions to go to the other side. Now with secondary active transport, this is when you use an exchange pump indirectly to drive the transport of other solutions. Long story short, you have your sodium-potassium pump, which is a method of primary active transport. Secondary active transport is going to take advantage of the primary active transport system to transport other solutions. So there's the symport system or the antiport system. Basically, symport system is two substances moving across the membrane in the same direction. So if your sodium ions are going in, well, why not we attach some glucose there and have them follow through? So that's a symport system. In an antiport system, the two substances are going to move across the membrane in different directions. So if you bring in a sodium ion, maybe another one's going to come out against the concentration gradient. So what should you remember for this lecture? We did talk about the structure and composition of lipids, which is very important. So know about the fatty acids, know about the glycerol that has to be attached to it to be considered a lipid, know about the different types of lipids and their function. Then we took a look at lipid membranes, understand the asymmetry of the lipid membrane, the fact that there's a fluid mosaic model that explains many different things, and the fact that your lipid membrane can have different domains and rafts that are specific to one type of protein and a function. We then moved on to taking a look at the types of function of those transmembrane proteins. And one of the main functions is transport. We were able to take a look at membrane permeability and potential, and also use Fick's law to model the diffusion of ions going through a concentration and an electric gradient. Apart from that, just know the difference between the different types of passive and active transport mechanisms, and then you'll be good to go.